everybody, welcome to my channel I Learn Daily. My name is Dr. Aman and I'm an optometrist. And today we're going to learn embryology of eye. In this video, we will learn how and when different structures of the eye develop and how the development of one structure of the eye stimulates the development of another structure of the eye. And in the end, we will discuss about some major developmental abnormalities of the eye. In order to understand the embryology of eye, we must first understand the basics of neuroembryology, as eye is an outgrowth or projection of the brain. Now, the embryonic central nervous system consists of four structures: prosencephalon, mesencephalon, rhombencephalon, and the neural tube. Now, prosencephalon consists of two subdivisions: telencephalon and diencephalon. Telencephalon, which will make the upper part of the cerebrum, and diencephalon will make the bottom part of the cerebrum. The mesencephalon will make the midbrain. The rhombencephalon also consists of two subdivisions. This right over here is formed from metencephalon and this over here is formed from myelencephalon. Metencephalon will make pons and cerebellum, and myelencephalon will make medulla oblongata. Pons, cerebellum, and medulla oblongata together makes the hindbrain, and this neural tube will make the spinal cord. The structure which is responsible for the development of the eye is diencephalon. Now, as we all know that an embryo develops from three germal layers: ectoderm, mesoderm, and an endoderm. The ectoderm subdivides into neuroectoderm and surface ectoderm. The neuroectoderm forms the central nervous system. The first diagram shows the superior surface of the diencephalon. The diencephalon, which is a part of the central nervous system, is made from the neuroectoderm and is surrounded by the surface ectoderm. During the third week of gestation, the diencephalon makes a projection from here and makes optic vesicle. The surface ectoderm from the center thickens and forms lens palacord. So we can say that during the third week of gestation, the development of the eye initiates. Now, the lens palacord makes an invagination into the optic vesicle, and the optic vesicle becomes the optic cup, and the lens palacord becomes the lens vesicle. This right over here is the optic stalk, which will later become optic nerve in future. During the fifth week of gestation, the surface ectoderm loses contact with the lens vesicle, and the lens vesicle purely lies inside the optic cup. The optic cup and the lens vesicle is surrounded by mesenchymal cells. Mesenchymal cells are derived from mesoderm and are a type of stem cells. The inferior portion of the optic cup is not fully closed, and the mesenchymal cells will form a hyaloid artery which will enter from here and will form a vascular lens capsule and will provide nutrients to the lens. The outer layer of the optic cup will make the outer layer of the retina, and the inner layer of the optic cup will make the inner layer of the retina. The outer 1/5 portion of the retina does not develop into neurosensory layer, rather it will stay as a single layer of columnar cells. Initially, only the outer layer of the retina consists of nuclear cells, so it will divide the retina into outer nuclear layer and the inner marginal layer, which is devoid of nucleus. Later, the outer nuclear layer sends its nucleus to the inner marginal layer. The outer nuclear layer nucleus will form horizontal cell nucleus, photopigmentary cell nucleus, which are rods and cones, and bipolar cell nucleus. The inner nuclear layer will form ganglion cell nucleus and amacrine cell nucleus. The inner marginal layer consists of cilia. This cilia later in the 7th week of gestation will be replaced by the outer segments of the rods and cones. During the 8th week of gestation, each nuclear layer of the retina can be differentiated. In the center, the ganglion cell starts to move to the periphery, leaving a central shallow depression, which is called macula lutea. After the birth, there is still present a single layer of ganglion cells, which is the reason why babies do not have a sharp central vision. This point right over here is called the optic stalk. The nerve fiber layers from the ganglion cells will travel towards the optic stalk to form the optic nerve. As we all know that the axons inside the globe is not myelinated, the optic stalk will provide the neuroglial cells and will make the optic nerve myelinated. The process of myelination also continues for a short term after the birth. The lens vesicle has been formed and now it consists of crystalline proteins. The single layer of cells which are present at its posterior surface will elongate to attach to the anterior surface. The nucleus inside each fiber will also travel towards the anterior surface, and we will have a nuclear bow at this point. Soon, as the lens fibers attaches to the anterior surface, the nucleus of these fibers disappear. These fibers are called primary lens fibers. Now, lens also consists of secondary lens fibers, and these fibers are produced throughout the life with the help of mitotic divisions with the help of cells which are present at the equator. Let's suppose this is the equator and these are the primary lens fibers. The secondary lens fibers will grow in this fashion. Now you can see that we have a Y suture over here, and its posterior surface will consist of an inverted Y suture. This suture is very important in phaco-chop and phaco-stop cataract extraction. This point is called ora serrata, the point at which the neurosensory retina ceases. The layer grows to the anterior surface and forms the pigmentary and sensory layer of the ciliary body. This layer will form the endothelial surface of the iris. This layer will form the constrictor and dilator muscles of the iris. The mesenchyme, which is present around the optic cup, will form the ciliary body and the ciliary muscles. It will also form the vessels and tissues of the iris. The mesenchymal cells are also responsible for the formation of choroid and sclera. The anterior surface, the epithelium of the cornea, is formed from the surface ectoderm. And the stroma and the Bowmans are built from the mesenchyme. The descemet is formed from the neuroectoderm. And the descemet is formed by the endothelium of the cornea. The primary vitreous consists of hyaloid vessel, which is providing nutrients to the lens. The secondary vitreous consists of type 2 collagen, which is formed by the retina. And the tertiary vitreous, the hyaloid vessel decay, and it consists of hyalurocytes and collagen. And in the end, the suspensory ligaments are formed. The eyelid is formed with the help of surface ectoderm. And the eyelashes as well, and the glands of mole and Z's. The oblique muscles, which are superior and inferior oblique muscles, are formed first from the mesenchyme. The recti muscles are first formed at the insertion, which are not actually differentiated. Later, they reach their cone and differentiated later at the insertion. The orbit is formed from the mesenchyme. Now let's discuss some developmental abnormalities of the eye. First comes coloboma. Coloboma is a condition in which any structure of the eye is underdeveloped. The second comes encaloblephron. Encaloblephron is a condition in which the upper and lower eyelid are not fully separated. And third one consists of persistent pupillary membrane. So this was all for today. I hope you all liked my video. I hope it helped you a lot. And till then, like and subscribe.